The story I'm going to tell you about today was led by this fantastic postdoc in my lab, Mark Plitt, in collaboration with Dan Turner-Evans at University of California, Santa Cruz, and with help from all of these fantastic folks here. And today, what I want to talk to you about is how the fly's brain flexibly links their internal sense of direction to external cues in their environment. And if you've ever walked to get a drink of water in the middle of the night and bumped into something, you probably appreciate that visual cues are really important for navigation. However, how external cues are incorporated into our brain's representation of our orientation in space is not fully understood in any organism. Furthermore, interestingly, learning your surroundings is interesting to me because it's an ongoing, what I'm going to call unsupervised learning process. If I asked any one of you to close your eyes and point to the door of the room you're sitting in, you could probably do that even without me having ever explicitly instructed you to learn that knowledge, or even regardless of the fact that the door may not have a positive or negative valence. But today I'm going to tell you about how in flies, this type of unsupervised learning process is regulated by multiple neuromodulatory systems and our progress towards trying to understand its synaptic mechanisms. So how do we study the sense of direction in flies? It was recently established that head direction is represented in this donut-shaped brain region here that sits in the middle of the fly's brain. And in this region, there are neurons called 𝗣𝗥𝗘𝗦𝗧𝗥𝗔𝗖𝗧 that have their dendrites that tile sectors of the ring with adjacent neurons representing adjacent angles of heading. And here I'm showing you a reconstruction from one of the two recent connectomes in the fly, showing each of these 𝗣𝗥𝗘𝗦𝗧𝗥𝗔𝗖𝗧 neurons labeled in a different color, so you can really appreciate this wedge-shaped dendritic structure. What this adjacent neurons representing adjacent angles means functionally is this. At any point in time, there's a single region in this donut with what we're going to call a bump of increased neural activity that fills a part of this donut. And over time, this bump tracks the direction of the fly. So for example, this wedge could be active when the fly was facing forward, but then like the needle of a compass, this bump tracks the fly's direction as it turns. And this offset between the fly's angle and the bump angle is actually arbitrary, and it can differ depending on the current external cues that the fly uses to orient. And we actually use this extreme form of plasticity to try to understand how neural representations like this are rapidly updated by sensory experience. By way of further introduction, let me show you a video of these 𝗣𝗥𝗘𝗦𝗧𝗥𝗔𝗖𝗧 neurons activity from their discovery by Johannes Selig and 𝗣𝗥𝗘𝗦𝗧𝗥𝗔𝗖𝗧. And while this video plays, watch how there's always one region of this video that is kind of, the field of view here is exactly the same as this donut I've shown in the cartoon. And they're doing GCamp imaging of this whole population of neurons. And I want you to notice that this GCamp fluorescence signal always resides, the peak of it always resides in one region of the donut. And while it may be able to go up and down in amplitude, it swings around, and it's swinging around tracking the direction that the animal is turning on this air cushion ball that is happening simultaneously. Importantly, these 𝗣𝗥𝗘 cells are head direction cells in flies, and they're very analogous to those that have been found now across the animal kingdom in many, many different organisms. And this suggests that head direction cells represent a kind of general solution for navigation, and that they're one core element that provides the brain with a relative internal compass. And this really motivates us because it makes us think that if we can understand the algorithms and how they're implemented for head direction cells in flies, it may inform more general understanding of spatial processing. And the thing that's really captured my fascination about this compass network is that it uses multiple forms of information to estimate head direction. This tracking can happen even in the dark by integrating self-motion information. However, in the dark, this compass will start to drift over time as errors build up about the exact step counts of its turns. Alternatively, however, when external cues are available, like visual cues, which is what I'll focus on today, but other cues can be used as well, this compass is often much more accurate when it has them, and it can track those cues very faithfully. And let me show you some example data from our lab that illustrates that point really well. So in these experiments, what is done is that the fly is glued under a 2-photon microscope, as we're showing in the schematic, and placed inside a virtual reality system comprised of a series of LEDs that can animate things around the animal. And then the fly walks on an air-cushioned ball, like I'm showing you in this video here. And that allows us to track this ball in close to real time and use that information to update the visual display around the animal. And for all the data I'm going to show you in the talk today, we always display a vertical blue bar that is locked to the animal's angular position to simulate a Q at infinity. You could think of it like the sun. So when the animal turns to the right, the Q will move to the left on its retina, or when it attempts to turn to the right, simulating the way the world would naturally fall on your eye if you were freely moving, but it's a virtual system. And then what we do, and by we, I should say all this data is from Mark Plitt, is Mark can measure the EPG neuron activity, and he does this by expressing the genetically encoded calcium indicator 𝗔𝗕𝗦𝗧𝗥𝗔𝗖𝗍 within this whole population of cells. And then he does volumetric imaging of this whole compass brain region here, this ellipsoid body region. And I'm going to show you a lot of plots like this. So let me unpack how we're going to plot this data. To plot this circular data, what we're going to do is we're going to cut this network open here anatomically at this pi region and unwrap it on the y-axis, and then display a heat map over time to show this neural activity. And so this is while a fly was walking in virtual reality with a single bright bar as a cue. So the first thing I want you to notice is that there's always one region of this network that has the darkest signal. This is the highest fluorescence. That's that bump. And that bump can move to reside on different angles in the anatomical structure as the animal walks. And if now on that same data, I overlay a red dot for each point in time that tells you which direction the fly was facing in virtual reality, I hope you can see that the fly's brain can track orientation very accurately. However, this visual bar is really critical for this accurate tracking, because if on the very next trial, Mark puts this same fly, continues to image, but removes the bar and has them instead walk in darkness, a couple of things happen. So look at this heat map on the lower half now, and I want you to notice a couple of things. First, I want you to notice there's still a bump of activity. There's still always one region in the network that's the most active. And when the animal makes large turns, now we're plotting what they did and where we would have animated the bar if it was on. You can see that the network activity can move in response to those maneuvers. But if you look at the longer time scale over a couple of minutes, I hope you can appreciate that while this network originally started out kind of pretty aligned with the virtual reality heading, by the time you get 3 minutes later, they're almost completely out of sync. And it's as if they've started to kind of drift and not be able to keep track on the time scale of many, many seconds. To quantify this drift, I'm going to use a metric throughout the rest of this talk that I'm going to define a value that I'm going to call the offset. The offset definitionally here is the difference between the neural heading, that's our estimation of where the peak of this dark region is, the angle of it, subtracted by the VR heading, the direction we think the animal's facing in VR. And for these two example trials, here I'm plotting the distribution of those offsets. And so I hope you can appreciate in the trial where these two things are moving around in lockstep, this distribution has a really clear, consistent peak. Whereas when the fly walks in darkness, the offset distribution often gets very flat. And this is because these two values are sliding past one another as the animal walks. However, so this really argues to the field, and we're just replicating data here, but I wanted to show you it with real data, that these EPG neurons, they really require these landmarks to stay accurate on the timescales of minutes. An important point to mention, though, because it's going to be relevant again in a moment, is that this example fly has an offset of 0, but this compass is actually a relative compass, and so this offset is arbitrary. So I can show you another example of another fly that still has very stable tracking. Watch how every time the animal turns, the neural activity rotates by the right amount corresponding, but these two values are now no longer on top of each other. And the offset in this fly is closer to about 180 degrees, or pi, almost completely opposite. And this is like many head direction cells in that these cells form a relative compass. And you already might have some sense intuitively of how to think about this, which is that when you're in a room, you may know your way around that room without having to know magnetic north. And it's still useful to orient yourself within that room and use a relative reference frame. And so that's how we think about this representation. And furthermore, not only can this offset vary from fly to fly, but this offset can also change within experiment, either following disorienting visual experiences or genetic visual pairing. And this has now been established. I think these data together really lead to the argument that the network's relationship with visual cues is pretty plastic and is plastic in adulthood in a way that can change quite rapidly even within an experiment. And this leads me to the core question of my talk that I want to address today, which is, what are the plasticity mechanisms that enable these external visual cues to be flexibly tethered to this internal spatial representation? To tell you about this, I need to introduce you to one other cell type. It turns out that visual information comes into this head direction network from a population of neurons called ring neurons. And these ring neurons have really beautiful anatomy. They have a dendritic arbor here, and then a ring-shaped axon that arborizes throughout the whole circle of this compass. Here's just a schematized version of that I'll use a lot throughout the rest of this talk. And we know now from connectomics that every single ring neuron synapses onto every single head direction neuron around this whole compass. And there's not just one ring neuron, there's many. Here I'm just showing you a small subset example of these. And these are visual neurons, and they respond stably to visual cues at particular locations in visual space. But as a group, they tile visual space, which sets them up really well to convey visual information into this network. And so let me take you through the working model for how this visual tethering might work. It's kind of the current working model in the field. Imagine when a fly first sees a landmark, like the sun, for example. Ideally, what you want it to do is you want this network to learn that the fly's view of that landmark correlates with the current direction it's facing, wherever this bump of neural activity is. It's hypothesized that this could happen by associative synaptic plasticity. And in particular, because these ring neurons are GABAergic and thus inhibitory, it's been proposed that this associative plasticity should actually be manifest as synaptic depression, or weakening of this inhibitory connection between the visual neuron and the currently active compass neuron. It's also presumed that there must be some type of non-associative synaptic potentiation to keep the general balance in the level of synaptic strength in the network. And once learned by some plasticity mechanism like I've laid out, then landmarks can correct for errors to make the compass do better. So for example, if the sun then temporarily disappeared and the fly got disoriented, if it saw the sun come back into view from that same angle, this synaptic association here could actually move the bump back and correct for the compass. And in this way, plasticity could enable the fly to start from a coarse compass that at first just relies on its own walking, on self-motion, but over time is refined using plasticity by storing information about currently available landmarks. And I would argue that the current published data in the field provides very good evidence for the general framework of this model, but that we still have very little information about the exact synaptic mechanisms of this plasticity. And so that's exactly what Mark has been working on. He wants to understand the mechanisms of this plasticity. And to do that, he's been asking the first question, which is, how do these synapses actually detect that they were active at the same time? How do they detect coincidence? To form an association, we think it would make sense for plasticity to be triggered by coincident pre- and postsynaptic activity, or cells firing together. So we've been starting to explore how this coincidence detection could be performed and regulated. We think of this as being able to happen in two core ways. Either coincidence could be sensed within the postsynaptic cell by some type of postsynaptic mechanism, presumably that would relate when neurotransmitter binds postsynaptic receptors and this postsynaptic cell being active. But because these synapses are GABAergic and these receptors pass chloride, it's a little less obvious to us how coincidence detection would happen in that manner. And this is especially true because we know that these cells don't express high levels of metabotropic GABA receptors, Alternatively, coincidence could be sensed in the presynaptic terminal by some type of presynaptic mechanism. And there we'd be thinking about some type of retrograde signal of some form that would carry information that the postsynaptic cell was active back onto this presynaptic terminal so that it could sense whether it was coincident with its own activity. Here, there's another puzzle, which is that flies don't have one of the most canonical examples of this type of retrograde signaling for plasticity, which is endocannabinoids. They're not existing in Drosophila, this famous retrograde signal. So we started to wonder, and we're really inspired by work in the insect mushroom body about whether neuromodulation, in particular monoamines, that input onto this presynaptic terminals could be useful part of such a retrograde signal. And part of the reason we were so interested in that idea is that it's been shown in the insect mushroom body that neuromodulation on presynaptic terminals can do can instruct synaptic depression. So it was really well set up by a framework, at least from a biochemical standpoint. So we wanted to explore, can neuromodulatory signals onto presynaptic terminals participate in retrograde signaling in a way that informs plasticity? It turns out there's a number of neuromodulatory cells that innervate these presynaptic visual terminals, and I'm going to tell you about two monoaminergic systems today. The first is a dopaminergic cell class called EXR2, of which there are 4 cells. I'm showing you their anatomy from the connectome. And the second is an octobaminergic cell class that I'll tell you a little bit more about in the moment. But to cut ahead to the punchline, these octobamine cells have wedge-shaped arbors, while the dopamine cells have more global arbors that are each cell innervates the whole compass region. And so we actually think that maybe octobamine cells are going to be the critical signal for synapse-specific plasticity, and that maybe dopamine is doing something different. But we wanted to test that a little bit more carefully. And so let me show you some recent data to kind of really dig into rigorously whether that's the case. Recently, what Merck's been doing is he's been using a dopamine sensor called 𝗣𝗥𝗘𝗦𝗧𝗥𝗔𝗖𝗍. He's been expressing it in these ring neurons so we can look at the dopamine drive onto these ring neurons while the animal walks in virtual reality. And he wanted to test this because while the dopamine arbors are broad, we wanted to rule out the potential that they may still have local release that is happening within individual wedges at a time, because that would actually be really well set up to instruct plasticity. But that is not what he's seen. He sees that dopamine release is extremely broad. Here, I'm showing you an example trial from one of our data sets where he's again unwrapped the neural data of this compass into its anatomical angles. And then we're plotting over time what the dopamine signal was at that particular anatomical location as the animal walks around and faces different directions. And what I want you to notice from this activity is that you can't see one region of bump of activity like you can in the other imaging data I showed you, but instead, every time the animal makes a rapid turn, there's a large flash of dopamine release that's global and seems to be across the whole brain region. I hope you can appreciate that here by looking at our plot of rotational speed over time and seeing that every time rotational speed peaks, we see a large global dopamine signal. And I just want to note that this dopamine signaling is positively correlated with the animal's fly's rotational speed across a large data set. And that is perfectly consistent with previous measurements done by Michael Marquis and myself using 𝗔𝗕𝗦𝗧𝗥𝗔𝗖𝗍 in my was a postdoc in Rachel Wilson's lab. So it really seems like these dopamine neurons are releasing dopamine from all of their terminals into the whole compass region every time the fly turns. And this is consistent with previous work. And because this dopamine story is published, I'm just going to summarize it very briefly. But basically, we think now that dopamine is maybe increasing its activity when the fly turns, and that this might be a useful gain on learning, but maybe not the proximal signal to do it. And both genetic activation and perturbation experiments that are previously published support this general framework. And I just want to take a moment to kind of answer a little bit of the why, which is, why would you want to learn more when you're turning? Why is that useful? And imagine the scene that I'm showing in this slide. If you're standing still and new objects appear in your view, let's say this flock of birds, it's very unlikely that those moving objects are useful orienting landmarks. But if you yourself are turning, then your view of the sun and these mountains and all these things that are in the distance in this image, those are going to move on your retina as you turn. And I'd argue that that information is critical. And so we're thinking that for the compass network, when the animal is turning is a really good time to learn, and that the dopamine's role may be more of kind of marking that favorable learning period than doing something proximal that actually allows for synaptic plasticity. Next, Mark turned to looking for what happens when we measure octopamine signaling. So now he did the same expression system, except instead of expressing the dopamine sensor, he expressed a newly published octopamine sensor in these ring neurons so that we can look at how these ring neurons feel octopaminergic tone. And I hope you appreciate that the neural data here is much different in that there was this bump of activity of octopamine release that moved to different regions in the compass. And if you look below at the animals heading over time in orange, I hope you can appreciate that this octopamine release signal is highly correlated with the animal's own heading within virtual reality as it walks around. And that's not just true for this example trial I'm showing you here, but across a much larger data set. If Mark uses the first half of the trial to find the best heading alignment, and then he applies that phase shift to the second half of the trial, you can see really robust alignment of this octopamine grab signal to the heading of the animal. And this is never the case for the dopamine signal that he's measured. We now know, thanks to Dan Turner-Evans and Tanya Wolf, who discovered the cell types that we think are the source of this octopamine. And when they found these cells, they were really intrigued by the wedge-shaped anatomy of these cells. See how each of these EL cells, or the names of these octopamine cells, tile wedges. And this tiling looks really similar to the tiling of the head direction cells, the EPG cells I showed you previously that I told you about. And this is intriguing because it turns out that the main input to these octopamine neurons, or one of their main inputs, are the head direction cells. And some of their main synaptic outputs are local regions of these visual axons that span this network. And this anatomy would allow that compass signal to be relayed locally onto a specific section of visual axon by traveling through the activity of these octopamine neurons. And this is a really clever way that the system could implement coincidence detection, because there would be a feedback loop from onto the presynaptic terminal, and that would get information about the postsynaptic activity. And the GrabOA data I showed you already argued that maybe these EL neurons are carrying a copy of head direction onto the visual terminals. But to test the extreme version of this idea, we're really predicting that these EL octopamine neurons have a copy that is matched in lockstep with the head direction cell signals. So to measure that, we turn to dual color imaging. For this, we performed dual color imaging where we measured the EPG activity using 𝗔𝗕𝗦𝗧𝗥𝗔𝗖𝗍, a green calcium indicator, and the octopamine cell activity using 𝗔𝗕𝗦𝗥𝗘𝗦𝗧𝗥𝗔𝗖𝗍, a red calcium indicator. And what we found is that these EL neurons, like the octopamine, also exhibit increased bump of activity that tracks head direction in lockstep. And importantly, each fly had nearly indistinguishable offset from the head direction cells or the octopamine cells. But importantly, these signals are not identical. And we've done a number of controls, so we're sure this is not an effective bleed through between the channels, but the fact that both of these neural representations are very similar and are always residing in the same region in the network at the same time. So this data argues that these octopamine neurons do in fact carry the relevant information that would be useful for learning and that it synapses with that information back onto the visual input synapses. So next we wanted to test whether octopamine was actually necessary for plasticity based on these ideas. To do this, we knocked down the synthesis enzyme for octopamine in these EL neurons by knocking down the enzyme tyramine beta hydroxylase using cell type specific driver developed by Tanya Wolf. And then during that knockdown, Mark measured the compass's visual tracking by comparing its activity in darkness to virtual reality with a single bright bar as a cue. And what we found is first that when control flies walk in darkness, they have really poor tracking or highly variable offset. But then when that same fly is given a visual cue in virtual reality, that offset improves and the tracking gets much better. However, when octopamine is disrupted, the compass tracking was indistinguishable whether the fly was walking in darkness or given a visual cue, which is to say, without octopamine synthesis, this neural activity is blind to the stimulus. And our interpretation is that this is because plasticity is disrupted. Next, we wanted to ask, are octopamine cells sufficient to instruct plasticity? To do this, we used localized two-photon optogenetics in order to activate these octopamine neurons in a single anatomical wedge at a time. We did this using spiral scanning with a 1064 nanometer laser and by expressing the optogenetic activator CS crimson specifically within these octopaminergic neurons. We're going to spiral on top of a single wedge within the ellipsoid body. In parallel, Mark also performed calcium imaging from the core compass neurons, these EPG neurons that express GCAM while the animal walked in virtual reality. And let me show you data from an example one of those experiments. First, let me show you a baseline session. Here again, the red dot is the cue angle of that bright bar while the animal walks in virtual reality. And this heat map shows the activity from the whole compass network unwrapped and plotted over time with the darker region showing increased activity, that bump. And we can see in this example fly that the bump is consistently offset relative to the cue angle. Here it's below by about 90 degrees. And when the fly turns and walks around and faces a completely different direction, the bump follows with a similar offset. You can see that here, you can see that here. These things are really moving in lockstep. Hopefully I can further convince you of that if I plot the offset between the compass angle, the brain activity, and the cue angle, this bright bar. And I can show you a histogram of that over a much longer trial. And you can see that it's highly peaked around slightly higher than 90 degrees. Then what Mark does is he pairs octopamine stimulation with a particular visual offset. And I'm going to show you what we're going to call a 0-degree offset. So what he does is he activates the octopamine neurons in a single wedge with a particular offset relative to a visual cue for a little under half a second, while simultaneously showing the animal a visual cue in front of them. Then he rotates the visual cue and the optogenetic stimulation sequentially by the same angular amount and goes around the circle all the way around 5 times. Then after that stimulation, we remove stimulation and return to imaging of the network to see if anything has changed. And following stimulation, Mark sees that the compass activity and the visual cue become almost completely aligned. Here, I hope you can appreciate that now the blue peak in this neural activity is right on top of the head of the cue angle, and they're following each other with a 0-degree offset. You can also see that that now the histogram is peaked almost perfectly at 0 degrees. So essentially, we've given the fly a new sense of direction by this short timescale perturbation using optogenetics. And the same fly, Mark can then play this game again, and he can remap the system again to 180 degrees. And now you can see that the distribution shifts to be peaked right at 180 degrees. And I was shocked when he showed me this data. This pairing works extremely consistently across flies with a very low error rate relative to the angle that we choose to target. And super importantly, this effect is associative. It requires these EL neurons to actually express the crimson opsin, and it requires the visual cue to be displayed for any remapping to occur. Now, it's important to note that in a previous study led by Sun-Soo Kim, 𝗣𝗥𝗘𝗦𝗧𝗥𝗔𝗖𝗧, and colleagues, had shown that you could actually remap this compass by activating the head direction cells themselves and pairing that with a visual stimulus. And if it's true that our model is correct, that octopamine is really the central mechanism of plasticity, then the prediction is that that pairing works because it recruits octopamine release. And so to test this question, we decided to first replicate this EPG neuron stimulation protocol on our microscope under our parameters. And we see exactly what was published by Kim et al., which is that this is also a very robust protocol, even under different parameter conditions. And next, we wanted to strongly optogenetically silence the octopamine neurons during the same experiment. And to do this, we've recently cloned a potassium channel opsin, 𝗔𝗕𝗦𝗧𝗥𝗔𝗖𝗧, into flies. This that was been published previously. And this is work from Ariana in the lab, first just confirming that this expression works well and that this is a good system for doing silencing of these neurons. Ariana performed patch clamp electrophysiology from these EL neurons, and they provided short pulses of light. And I hope you can appreciate that a cell that was going along spiking at a pretty depolarized potential then gets robustly, rapidly hyperpolarized with a short pulse of light. And this is the case not just for this example trial, but across a much larger data set. And what's made us really excited about 𝗔𝗕𝗦𝗧𝗥𝗔𝗖𝗧 is actually that it has a really ideal range of 2-photon absorption, right? Kind of similar to the laser line we've been using for crimson. And so what this allowed Mark to do is to go back and replicate the same remapping experiment that Kim et al. Did, where crimson is expressed in the EPG cells, but now in parallel, using a different expression system, we have expressed 𝗔𝗕𝗦𝗧𝗥1 in the octopamine neurons. And what this means is that while that 1064 laser line is spiraling, it's both activating the EPG neurons, and it will also be locally inhibiting the octopamine neuron in that same region. And what we find is that when we do this silencing, we occlude the original learning effect that we can see by just activating EPG, arguing to us that these EL neurons are really critical for all of these forms of plasticity that have been reported. So let me summarize what I've told you in this talk today. I've told you that plasticity enables flexible visual head direction mapping. I told you that we find that there's a highly motor-locked dopamine signal that we think is important for shaping plasticity, but that might not be the critical signal to actually perform coincidence detection. And then we talked a lot about this octopamine cell type that we think is really critical for this process. We find that octopamine neurons relay a copy of head direction back onto the presynaptic visual input terminals. We show that if we perturb octopamine synthesis by knocking down its enzyme, that it's necessary for visual tethering, and we can break the compass's ability to reference to external cues. And then finally, I showed you data at the end that argues the idea that the activation of these octopamine neurons is really sufficient to instruct plasticity. And taken together, these data lead to our current working model, which is that we think coincidence between a visual cue and head direction is detected in these presynaptic terminals when octopamine binds to local receptors that are expressed here, and these ring neurons are coincidentally active, and that this leads to some plasticity mechanism that changes neurotransmitter release in some manner. And there's two core principles that I want you to take home from this talk. First, we're really excited about seeing coincidence detection at an inhibitory synapse being solved by relay through a third neuromodulatory cell. I cannot find an example of this exact synaptic motif in the literature, but I think that it's a useful motif that could definitely exist in other systems now that we've discovered it. And yet, while this motif of kind of taking a copy here and relaying it back to enable plasticity and inhibitory synapse is quite unique algorithmically, it's performing coincidence detection between pre and post by a third player. It shares a lot of similarities to plasticity mechanisms that happen at excitatory synapses in the insect mushroom body. The second thing I want you to take home from this talk is that neuromodulators like octopamine and dopamine can do unsupervised learning also. So I think monoamines, especially dopamine, have been really famous for their role in signaling reward or punishment in order to instruct learning. But for spatial learning, we're seeing these same monoamines act at different levels of abstraction. Here, in this case, with octopamine implementing coincidence detection, we think, and with dopamine maybe acting as a wider gain on the system to tell the circuit when it might be best to learn. And with that, I'd like to thank my lab and all the people and funding that contributed to this body of work. Again, all the data I showed you today was collected by Mark Plitt in my lab with help from Jessica Ko, Ariana Layden, Dan Turner-Evans, and 𝗣𝗥𝗘𝗦𝗧𝗥𝗔𝗖𝗍. And here is us being very good wearing our safety glasses in front of our Ultima 2P Plus that collected almost all the data that I showed you today. And thank you, and I'd be happy to take it.
Yeah, thank you so much, Yvette. Very cool story. We have a couple questions. First one came in earlier. Are the head direction to octopamine synapses 𝗥𝗘𝗥𝗧𝗥?
Are the head direction to octopamine synapses 𝗥𝗘𝗥𝗧𝗥? So the thing that's interesting in flies, I have a couple of things to say about this. So yes, in some sense. So in flies, some cells are really polarized and have like regions of their arbor that you can really carefully define and say this is definitely an axonal region, and other regions that are like really more predominantly input regions and are dendrites. I'd say that this region of the ellipsoid body, specifically for these head direction cells, for the EPG cells, are an exception. The arbors there are super input-output. Like people are call them dendrites, but they have a ton of output synapses. And so this is kind of a definitional question. If you want to call those axons, then these are axonal. If you want to call those dendrites, then this is technically dendroaxonal. But maybe it's just because that definition is too strict for this compartment. But it's definitely the case that they're going right on to the presynaptic kind of the output regions of these octopamine cells. Thank you for that question.
Yes, okay. We have a second question. How was the remapping done to go back to the original offset during the opto stimulation plus visual cue? And then a clarification where they said you have to go back from 0 degrees to the original like 80 degrees, etc.
Yeah. Sorry, I couldn't catch you there, Jimmy. You're asking me to go.
Yeah, they asked how was the remapping done to go back to the original offset during the opto stimulation and visual cue. For instance, to go back from 0 degrees to the original 80 degrees.
Yeah, so in this plot here, which I believe is what the audience member is asking about, what we did is we did a baseline imaging session, 6 minutes of the animal walking where we're just doing GCamp imaging to see the compass. And then we pause and we do this 2-photon object activation pairing. The first time in this example that Mark did it, he did a 0 degree offset pairing. And then here I'm showing you a subset of an example trial, but that we actually record for 6 minutes. And that's what goes into this distribution of what happened after that pairing. And then what I haven't shown on this slide, but is the case and is leading to the next histogram, is then we pause. And this time we do a mapping where when the bar is in front, we put the optogenetic activation to the bottom wedge and rotate those together. So now they have a different phase offset. And what happens when we do that, and if I had the data here to show you, you would see that the bump then starts to avoid this head direction region by about 180 degrees. And then that is what I'm reporting here in this kind of pink color with the 180 degree opto offset. I hope that answers this individual's question, but they can follow up if not.
Sure. Yeah, we'll give them contact info for the follow-up if needed. I had one question. I was wondering, do you see any translational opportunity down the road by fixing this compass mechanism, improving plasticity in implements?
Yeah, it's a great question. So a couple of thoughts. I think you could think of like a proximal benefit to this work, which is like by understanding how spatial navigation systems work, if the way it works in flies is similar or some of the principles of the way it works in flies, whether or not it's like all the gory details, the implementation is useful in flies, that can then inform how spatial navigation may be breaking in humans where they have problems with this. But I guess I see this work as a more kind of basic science approach, which is that the reason I'm so drawn to head direction is not per se because I love spatial navigation, but because it's a really tractable way to ask, how do animals hold an idea in their mind, and how do they update this idea, and how do they keep this idea that is both an internally generated concept, but that also needs to kind of flexibly learn and reference the outside world? I'm really interested in the interplay about how those processes are implemented and regulated. And so I think the thing that I think is maybe the most human health relevant, though there are other relevance of this work for kind of robotics, for example. But the most human health relevant findings from these work, I would argue, is the kind of push to start looking more broadly at the role of monoamines in all kinds of learning, including unsupervised learning. And that could be spatial learning, but that also could be conceptual learning in higher order organisms. And I think the work we've pointed out here is just kind of a nice first example of the idea that you can repurpose these kind of canonical monoamine systems that people have typically studied in the context of kind of a reinforced learning system. And that now we're starting to find evidence that these are being used by the brain under much less reinforced regimes of learning. And I think, I would speculate that that could definitely be the case in larger animals and humans as well, and that we should take a second look at that.
That's it. Maybe one final question that we just enough time for is a question about what happens to the bump of activity if the animal does not move for a while as if when sleeping?
Two good questions. So I don't yet know about sleep. I know there are some groups working on this, but stay tuned. So I'm not going to comment on sleep per se. But one thing that's interesting that's been shown from its first observation of this bump of activity is that when the animal stops and stands still, and let's say they stand still in darkness, this bump of activity, it lowers in amplitude, but it doesn't go away. So it's still there for as long as anyone I know in our lab as well has been able to track it. So really arguing that the brain still has some internal representation of where the animal is facing. And that's exactly how we're thinking of it, as kind of a persistent internal representation of the animal's best guess of which way it is at that point in time. And the details of how that works are like a fascinating topic that my lab's interested in for sure, but that I don't have an answer for today.
Sounds good. With that, I'd like to thank you again, Yvette. Great talk, great story. Appreciate your time.